Welcome, everyone. Before we start, just want to say thank you for your participation today. We're well aware of how important everyone's time in lab is, and we appreciate your time away from it. So I wanted to introduce Kristen. She's a senior scientist in scientific affairs at Arima Genomics and holds a PhD in biology. At Arima, she spearheads quite a lot of our research and development initiatives, and it's been a pleasure to work with Kristen. She was recently with us at ESHG out in Vienna, Austria, a couple of weeks ago. Great to work with her there. And Kristen, I just wanted to hand the floor over to you.
All right, thank you, Christian, for the introduction, and welcome, everyone. It is my pleasure to be here today to introduce you and share some of our latest research from Arima Genomics. All right, so if you are new to the Arima community or to 3D genomics more generally, then welcome. We are a 3D genomics company based in San Diego, California, and at Arima, we provide kits as well as end-to-end services on a wide range of sample types. This allows our customers to use our core Arima Hi-C technology in order to address a variety of fundamental biological questions. So whether you want to try 3D genomics in your lab or want our help preparing and sequencing samples, then we can support you in either of those endeavors. So what exactly is the 3D genome? Well, to understand the 3D genome, let's start with what we're all familiar with, what we might call linear genomics. And we can define linear genomics as this process of figuring out the order of each of the approximately 3 billion A's, G's, C's, and T's in the genome in ideally one or maybe several straight lines. But of course, there's far more complexity to the genome than can be easily captured in the linear paradigm. What we know now is that the DNA is not just randomly shoved into the nucleus of cells. There's really a defined pattern to how chromosomes are packaged into compartments. And this is what 3D genomics allows us to describe. 3D genomics takes into account not only the sequence, but also the structure of how that DNA is organized in the nucleus to better understand gene regulation and genome function. So when we think about 3D genomics, we can see that the sequence is really just the tip of the iceberg when it comes to understanding genome function. And using a 3D genomics approach, you can understand not only the sequence, but structural variants like translocations, indels, inversions. And going further, it helps us understand mechanisms of gene regulation and even broader genome organization like chromosomal compartments, topologically associated domains, and looping structures, all of which can affect genome structure and genome function. So the types of questions that we can address with 3D genomics have applications in a lot of different areas of active research. 3D genomics allows us to describe the spatial organization of chromosomes, and this has contributed greatly to genome assembly, including the recent publication of the first T2T human genome. 3D genomics has also had an enormous impact on epigenetics, as it captures how spatial and temporal changes in chromatin conformation can alter gene regulation and cellular function. But the real promise of 3D genomics is that we can leverage these structural and functional insights from the 3D genome and apply them to major challenges in human health, with the goal of being able to understand the mechanisms underlying disease and ultimately of developing new therapeutic approaches. So let's take a look at an example of how 3D genomics provides key insights to understand and treat cancer. So this is a really exciting paper that was published in Nature Genetics in 2020 by Cloken and colleagues. And what they were able to demonstrate in this paper was how the 3D organization of an oncogene relative to its enhancer could affect the progression of T-cell acute lymphoblastic leukemia. So in this first set of diagrams representing a healthy patient, we can see that the oncogene, in this case it's MYC, and this putative enhancer element are approximately 2 megabases away from each other in linear space. And 3D genomics shows us that these two elements normally arrange into 2 individual looping structures that are very distinct from one another. However, in patients with this leukemia, we see a new transcription factor binding activity at that enhancer and a corresponding activation of the MYC gene 2 megabases away. Taking a 3D genomics approach, we can see that there is a rearrangement of these chromatin loops, and that leads to the enhancer element being brought into close proximity to the MYC gene, causing that aberrant expression at MYC. So the authors of this study went on to show that by inhibiting the transcription factor bound to this enhancer with pharmaceuticals, they were able to restore normal expression of MYC. So using 3D genomics, they were first able to identify the mechanisms by which MYC is misregulated in T-cell acute lymphoblastic leukemia. And secondly, maybe more importantly, inform exactly which transcription factor is needed to be targeted by the treatment in order to restore normal function. And cancer research is far from the only area in which 3D genomics is having this type of impact. The breadth of disease states that 3D genomics is starting to inform is really exciting. As you can see highlighted here, there are new publications coming out from immunology, from cardiology, neurobiology, developmental biology. So as you can see, 3D genomics is having a big impact in diverse fields relating to human health. So in the last few minutes today, I'd like to highlight how Arima's technology has been used to address these questions in human health. In particular, I want to focus on how we've identified novel structural variants in cancers. But first, just to give you a sense of how we visualize Arima Hi-C data, this is a heat map used by the T2T consortium to scaffold the complete human genome sequence. And what you're seeing here is each of the chromosomes lined up across the top and the side of this heat map. And each point on the heat map represents physical interactions between two regions of the genome. So the dark red points that you see along the diagonal indicate that there are a lot of these 3D interactions between those regions. And the light red or white indicates minimal 3D interactions. So as you can see, it's very common for chromosomes to form these 3D linkages within themselves. But in healthy cells, chromosomes rarely form these linkages with other chromosomes. So now let's look at the differences between a normal karyotype and an abnormal karyotype from a cancer cell line. So on the left, I'm showing again a normal cell line, and we see again that these interactions are primarily intrachromosomal. That is, they're interactions within the same chromosome and not typically between two different chromosomes. By contrast, in the cancer cell line, we can see this very distinctive off-diagonal signal. So each one of these squares is highlighting a 3D linkage or a structural variant between chromosomes that is not present in the normal sample. And these interchromosomal interactions are very characteristic of translocations. We can imagine here that genes A and B have been brought into close proximity because of the translocation. And what we've observed on the heat map are the 3D linkages that are now spanning this breakpoint that fuses the two chromosomes together. Finally, although we've highlighted translocations here, I want to point out that other classes of structural variation can also be identified from their signatures in Hi-C data as well. Using this 3D genomic information, we've undertaken a large pan-cancer benchmarking study with collaborators at Scripps MD Anderson, Children's Mercy Hospital in Kansas City, and NYU Langone to look at FPE samples across 20 different tumor types that have an archival time period of anywhere between 1 and 12 years. And to date, we have examined at least 165 patient samples and counting. So we have a lot of very exciting data coming out of this study, but as my time is very short today, I want to take you just through one example that's particularly exciting to me. So our collaborator, Dr. Matthias Nuderl at NYU Langone, had a pediatric patient with a stage 2 glioma that was initially treated with subtotal resection of the tumor. But 6 months after her surgery, this patient experienced a very rapid progression and the tumor returned quite aggressively. So NYU performed comprehensive DNA and RNA sequencing of the primary tumor, which was inconclusive. They found no driver mutations that they could see, and the correct treatment for the patient was kind of unknown still. So we performed a Hi-C analysis using Arima's Hi-C technology, and that revealed a novel structural variant on chromosome 9 near the PDL1 gene. So PD-L1 encodes a protein that helps immune cells recognize normal, healthy cells in the body. And in some cancers, high expression of PD-L1 prevents the immune cells from recognizing and targeting cancer cells, allowing those tumor cells to proliferate. Since the high-C data implicated this possible role for PD-L1 in this patient, our collaborators at NYU decided to look further into that result. And in fact, for this patient, the Arima high-C result was confirmed with immunohistochemical staining, which showed strong and diffuse PD-L1 expression in the tumor cells. So once we confirmed the PD-L1 expression, the patient was then started on immunotherapy with the drug pembrolizumab. Since that change in treatment, the patient has responded very well and her disease is now stable. So what's particularly exciting about this case to me is that by using 3D genomics, we were able to identify this novel structural variant that was missed by other genomic tests, and which really provided the key functional insight that enabled the medical team to prescribe the best treatment for this patient. Although this is just one example that I've walked you through, what we're seeing with our pan-cancer benchmarking study is that 3D genomics can really help cancer researchers in three key areas. So first, 3D genomics can help us detect known fusions, such as those involving NTRK, ROS, and these other really key cancer genes, as well as novel gene fusions. 3D genomics allows us to find the key gene fusions and structural variants that might be missed by other technologies, as we saw in the PDL1 glioblastoma patient. And finally, 3D genomics has really helped to unlock access to fusions from archived FFPE specimens, which typically provide few answers in molecular assays, especially for the older specimens. And we have demonstrated concordance with FISH and other methods in 10-year-old archival samples. So in conclusion, I hope I've helped convince you that there's much more to the genome than just the sequence, and that by using a 3D genomics approach, we can help provide novel insights for disease researchers. I hope I've also convinced you that Arima's high-C technology is a robust and reliable solution that is currently being implemented across a wide range of basic and translational research areas. And we'll see other examples of this in the next talk. So with that, thank you all, and I will turn it back over to Christian.
Perfect. Thanks, Kristen. Really appreciate it. Great talk as always, and really appreciate your talk at ESHG as well. Dr. Gisela Orozco is a Wellcome Senior Fellow in the Center for Genetics and Genomics versus Arthritis at the University of Manchester. Her main research interest is in the genetics of complex diseases, with a focus on using functional genomics to understand how genetic variants affect genes and biological mechanisms that drive disease. Sorry about the muting there, Gisela. Welcome and thank you. The floor is yours.
Thank you, Christian, and thank you for inviting me. Yeah, so good afternoon, everyone. As you all know, autoimmunity arises when the immune system fails to distinguish self from non-self. Autoimmune diseases are quite common and affect millions of people worldwide. Epidemiological studies have estimated that they affect between 3 to 5% of people worldwide. These are chronic diseases with no cure, and although there is a range of therapies available to alleviate symptoms, many patients do not respond to them. So it is clear that we need to identify novel therapeutic targets for this unmet clinical need. But to identify novel therapeutic targets, we need to understand what causes disease. So we need to understand what biological pathways are affected in disease so we can target them with appropriate drugs. But unfortunately, our understanding of disease etiology in autoimmunity is still quite limited. On a very basic level, we do know that autoimmune diseases are what we call complex traits. These diseases arise from a combination of multiple environmental factors and multiple genetic factors. Genetics is a key factor influencing susceptibility, And therefore, understanding genetics of disease can help us understand disease etiology. It can help us predict disease risk and predict treatment response. And also, it can help us identify new therapeutic targets. So how can we study the genetics of these complex traits like autoimmune diseases? Well, we can identify genetic variants or SNPs that increase disease risk by performing genome-wide association studies, or GWAS. We can take DNA from large numbers of patients and healthy people, and we can compare how frequently a particular SNP or genetic variant occurs between these two groups. If there is a variant that is present in significantly higher proportion of patients compared to controls, we say that that SNP is associated with disease. And we do this comparison across the whole genome for millions of SNPs. For the past 15 years, genome-wide association studies have been very successful in identifying multiple genetic regions or loci that are associated with risk of many autoimmune diseases. So this is just an example. In our lab in Manchester, we have contributed to the identification of a large number of risk loci for autoimmune diseases like rheumatoid arthritis, juvenile idiopathic arthritis, and psoriatic arthritis. But the ultimate goal of genetic studies is to improve human health, obviously, by applying these GWAS discoveries into the clinic. But so far, the translation of GWAS findings into the clinic and their use in drug discovery programs has been quite limited. And this is because it's difficult to assign causal genes to these genetic associations. Let's talk about this challenge in the post-GWAS era in a bit more detail. So a limitation of GWAS that prevents clinical translation is that genetic signals are difficult to interpret functionally. The vast majority of risk loci contain multiple highly correlated genetic variants or SNPs, and most of them map to non-coding regions of the genome. They do not map to proteins. So it's difficult to know which are the causal variants, what is their function, what gene or genes they have an effect on, and in which cell types. Effective translation of GWAS findings into improvements in healthcare requires answering these questions, but GWAS alone are insufficient to answer them. So how can these non-coding SNPs that map to non-coding regions, how can they influence disease biology? Well, we think that the most likely mechanism by which they cause disease is by altering the regulation of genes that are important in disease. There is now a lot of evidence showing that GWAS SNPs are enriched in enhancers that are active in cells that are important in disease biology. Just to remind you, enhancers are short regulatory regions that can regulate the transcription of their target genes. These are short non-coding regulatory elements that can turn the expression of genes on and off. And they are characterized by a number of features, including open chromatin or lack of nucleosomes, specific histone modifications like H3K for ME1 or H3K27AC, the binding of tissue-specific transcription factors, etc. These enhancers can be located at large distances from the genes they regulate, but they can come into physical contact through chromatin interactions or chromatin looping. So we can study these enhancer signatures, chromatin interactions or chromatin looping, and gene expression to functionally characterize GWAS loci using an array of functional genomic techniques that I'm going to explain briefly now. So here you have a cartoon of a typical GWAS locus. Each dot is a SNP, the highest, the minus log p-value, the most strongly they are associated with disease. And there are multiple associated SNPs that are highly correlated, or they are in high linkage disequilibrium. so we don't know which SNP is the causal one. And they map in a non-coding region between gene A and gene B, so we don't know which gene they might have an effect on. By looking at open chromatin in relevant cell types with a technique called ATAC-seq, we can identify what regions of the chromatin are active. And using a technique called ChIP-seq, we can map histone marks that are correlated with enhancer activity, as well as transcription factor binding. So putting all these complementary data together, we can infer that disease risk at this locus may be mediated by a disruption of the regulatory activity of this enhancer that I have highlighted here in red. And we can also narrow down which variant is the causal one. But what gene is this enhancer regulating? To identify disease genes, we can detect chromatin loops between disease-associated SNPs and genes using a technique called Hi-C. Traditionally, GWAS variants were assigned just to the closest gene. In this example on the screen, gene A is closer to the disease-associated variant in the linear view. However, gene B, which is further away, is interacting with the SNP in a chromatin domain, and gene A is outside this domain. So we can infer that actually gene B and not gene A may be important in disease. And we can also look at gene expression and see whether the transcription levels are correlated with the presence of the risk allele. In this example, the disease SNP is an eQTL for gene B, but has no effect on the expression of gene A, suggesting that gene B may be the causal gene at this locus. So these are a few of the functional genomic techniques that we use in our lab in Manchester to try to functionally characterize GWAS loci and identify causal genes. So our group and many others all over the world have applied all these methods to systematically characterize GWAS loci for lots of autoimmune diseases. We have performed lots of experiments in cell lines, and all this data has shown that we can use these techniques to identify novel disease-causing genes and pathways. We have seen how risk SNPs often do not interact with the nearest gene, but with genes that map at a long distance. So this is quite relevant for the interpretation of GWAS loci. We have also realized that there can be multiple potential causal genes and SNPs per loci, so everything is very complicated. It's not one SNP, one gene. We have also learned that small changes in chromatin interaction intensity that can be mediated by disease-associated variants can lead to large functional effects in gene expression. These effects are often cell type and stimulus specific. And all this data can help in the identification of novel drug targets and drug repositioning. So this is all good data, but as I just said, the regulation of gene expression is highly context specific and can be different in different cell types after different stimulus and in different disease states. And this is on the right, you have an example of how chromatin interactions can be different in GERCATS, which is a CD4 positive T cell line, compared to primary CD4 positive T cells isolated from blood. We have estimated that around 20% of the chromatin loops are different. Therefore, to understand the role of disease SNPs in the regulation of gene expression, we need to study primary cells from disease-relevant tissues isolated from patients. But this is challenging due to the limited availability of biological material from patients. Previous Hi-C methods that we had been using needed a high number of cells in the order of tens of millions. So it was difficult to apply these to primary tissues from patients. So when Arima kits became available, it was a very good alternative for us compared to homebrew methods, because these kits allow us to work with a low number of cells. And also the Arima method offers a much better resolution than previously available methods. And now I'm going to show you an example of a project that we have recently completed in systemic sclerosis using Arima Hi-C kits to identify disease-causal genes. So this is one of the autoimmune diseases that we study in my lab, systemic sclerosis or scleroderma. This is a complex chronic autoimmune disease that affects the connective tissues. It is characterized by an immune imbalance, vascular alterations, and an excessive collagen deposition leading to fibrosis. There have been several GWAS studies performed in systemic sclerosis, and 27 risk loci has been identified. But like in other complex diseases, most of these genetic associations are non-coding, and the causal genes are unclear. So to try and identify scleroderma causal genes, we isolated CD4 positive T cells and CD14 positive monocytes from blood collected from 10 patients with systemic sclerosis and 5 healthy individuals. And we chose these cell types because they are thought to be very important in the pathogenesis of systemic sclerosis. Using these cells, we performed whole genome promoter capture Hi-C, and this is a modification from the Hi-C protocol. In Hi-C, we detect all the interactions that are happening in a cell in a genome-wide manner. But in promoter capture Hi-C, we enrich the Hi-C library with biotinylated RNA baits that are complementary to all gene promoters in the genome. So in this way, we have a better resolution for the chromatin interactions that are happening at promoters. And we also did RNA-seq on all these samples, and we genotyped all the individuals involved in the study. So in agreement with previous reports, we found that chromatin conformation is highly cell type specific. And importantly, we see that differentially expressed genes are between the two cell types are significantly enriched in differentially interacting genes. So this indicates how chromatin conformation is highly linked to gene expression. And these results show that it is important to use the right cell type when using them to link variants to genes using chromatin conformation. In contrast, we found that 3D chromatin structure is largely preserved between patients and healthy controls. And it has been shown that subtle chromatin conformation changes can lead to big changes in gene expression. But these subtle differences in chromatin conformation can be difficult to detect. So we think that our study was perhaps underpowered to detect these subtle differences. And also there may be too much variability between patient samples. So we are working very hard now in the lab to try and work a little bit more on these differential chromatin interactions between patients and controls. And we are using Arima Hi-C kits to perform, to produce Hi-C map in a large cohort, in a larger cohort of patients and controls. And we are currently analyzing this data. Next, we used our data sets to assign potential causal genes to scleroderma risk variants that had been previously detected by GWAS. We identified 39 potential candidate genes using chromatin interactions, and we confirmed 7 genes which had been associated to systemic sclerosis just using genomic proximity out of a total of 46 genes that we identified using chromatin conformation. Interestingly, some previously associated genes in systemic sclerosis like IRF8, STAT4, or CD247 showed cell type specific interactions. And I'm now going to show you a couple of examples. This is the IRF8 locus that is associated with systemic sclerosis, but also with many other autoimmune diseases. These are the GWAS associated SNPs that are shown here in brown, and I have also highlighted the region in red here. And these SNPs are located downstream the IRF8 gene, and they overlap enhancers in T cells and in monocytes. So I'm showing results for T cells in blue, and monocytes are shown in red. So these SNPs only show chromatin interactions with the promoter of IRF8 in monocytes, but we couldn't detect any interactions in T cells. And transcriptomics showed expression of IRF8 in monocytes and not in T cells. So this data suggests that the risk variants that are associated with scleroderma at this locus may be contributing to disease by an altered regulation of IRF8 expression in monocytes, but not in T cells. Actually, IRF8 is a transcription factor that is very important in the differentiation and regulation of monocytes and macrophages. It's been shown that variants in IRF8 are associated with monocyte counts across different populations. And it's been reported that scleroderma patients have a down-regulation of IRF8 in monocytes that can affect the fibrotic phenotype of the disease. So this confirms that IRF8 is an important gene in systemic sclerosis. The STAT4 locus is another region that is implicated in lots and lots of autoimmune diseases, including scleroderma. The associated variants at this locus map to introns of the STAT4 gene, and they interact with the promoter of STAT4 only in T cells, where it is preferentially expressed and not in monocytes. And STAT4 plays an important role in T cell signaling and differentiation. But very interestingly, our data suggested that there could be an additional potential causal gene in this locus, NABP1, in both T cells and monocytes. This is a gene that is involved in DNA repair, and it had not been thought to be implicated in systemic sclerosis before. So this shows how chromatin conformation can suggest novel genes in autoimmunity. So this is another example where we can detect multiple novel genes in a GWAS associated region. So this is an example of associated variants in chromosome 11 that are located around the DDX6 genes. And we detected chromatin interactions between the SNPs and the promoter of DDX6, but also other potential candidate genes, including CXCR5 and others, as you can see in this region. And CXCR5 could potentially be very interesting in systemic sclerosis. This is a chemokine receptor that plays an important role in the differentiation of T follicular helper cells. And these cells have been shown to be increased in systemic sclerosis, and they are correlated with severity of disease. Finally, I would like to show you how we can also use chromatin interactions data to identify novel therapeutic targets. So we took the 46 genes that were linked to GWAS SNPs in our Capture High C data, and we looked at drugs that target either them or genes that show strong protein-protein interactions with them. We identified a total of 21 drugs that target 13 of the systemic sclerosis genes that we had identified using High C. 2 of these drugs, tocilizumab and nintedanib, are already approved by the FDA for its use in scleroderma. And we also identified 4 drugs that are currently in advanced clinical trials in systemic sclerosis. So this is a good positive control that indicates the validity of our method, because we are picking up drugs that are already in use for scleroderma or in advanced clinical trials. But we also identified 15 drugs which are already in use in other similar autoimmune diseases that could be repurposed for treatment in scleroderma, for example, metformin or dimethyl fumarate. So this study that I have shown you today is just an exemplar of how we can use 3D conformation and functional genomics in general to understand etiology of complex diseases. And obviously, this is a pipeline that can be applied to any disease with a genetic component. So we have similar ongoing projects in rheumatoid arthritis, psoriatic arthritis, juvenile idiopathic arthritis, also in asthma, osteoarthritis, psoriasis, even monogenic diseases. So in summary, genetics and genomics have the potential to inform drug repurposing by identifying the disease genes and biological pathways that we need to target. Most risk loci identified through GWAS do not map to protein coding genes and are difficult to interpret. And it's still early days, but using functional genomics, 3D chromatin conformation, bioinformatics, and data integration using disease-relevant cell types, this will all help in the translation of genetic findings into patient benefit and drug repurposing. Finally, I would like to thank all the patients and healthy volunteers who donated samples. Thanks to all the researchers that have contributed to this work, to the funders for their support. Thanks, Arima, for inviting me to talk to you today, and thank you for listening. I'll be happy to answer any questions if you have any. Thanks. I'm going to stop sharing my screen.
Perfect. Thank you, Gisela. Really great to see you again after ESHG, and always a great story. I love the layers of information that you share, and I probably have a few questions about how you go about doing that. We have a few here and a few more coming in, which is great to see. Let's start here towards the top. So this came, I think, right at the end of Kristen, your talk. I'll just read off the questions so everyone can hear it. And then Kristen, would be great to get your insights. I think there's really just maybe just going back a couple of slides to explaining the interpretation of heat maps from Hi-C. The question is, which kind of structural variation did you find in the patient with glioblastoma? From the chromosome 9 versus chromosome 9 read heat map, I cannot see that there was a translocation. Could you explain in detail the interpretation of the heat map?
Let me just share that slide again so we can look at it. All right, can we all see that?
We can, yeah.
Perfect. Yeah, so that's a good question, and good eye. This is not actually a sort of traditional translocation like between two chromosomes that we might have what I was alluding to in the introduction to this. This is definitely not like that. What we're looking at here specifically is all of these increased 3D interactions, and that in this case, even though it's not a like traditional translocation, it is still representative of this rearrangement. And what was really key to this particular study is just the fact that it was near PDL1. That kind of tipped us off that maybe something was interesting there, but the Hi-C results really weren't like the smoking gun on their own. They just led us to looking at the immunohistochemistry, and that was really the confirmation here. But yeah, you're right, that's not strictly a translocation, but yeah, I hope that answers the question.
Perfect. Yeah, I know these heat maps, especially via Zoom, might be tough to interpret a little bit. So great for the appreciate the background explanation. Let's see, scrolling back up to the top. I think there's a couple of questions about maybe for you, Gisela, about kind of study design. It looks like a little bit. There's a question here. So I would like to do a functional study of mRNA SNPs in rheumatoid arthritis. I'd like to know which cell line can be used for this. Any insights there? Oh, you're muted.
Yeah, sorry, I was muted. Sorry, 2 years of Zoom. Yeah, thanks for your question, Jasir. So there's lots of bioinformatics that you can do using publicly available data sets to understand which cell type you need to study for your specific research question. So what we have done in the past is to see in which cell types genome-wide association studies SNPs are enriched in active enhancers, because our hypothesis is that these SNPs will be regulating gene expression in cell types where these enhancers are active, obviously. So by overlaying your regions of interest, in case it's SNPs that overlap microRNAs, I believe, so you could overlay functional genomics, the epigenetics data sets, for example, from the EpiMap project. There's tons of publicly available data sets that you could interrogate that other people have done in lots of different cell types. So you can overlay your SNPs with these data sets to see in which cell types these regions are active. And that can help you identify the cell type that is more interesting for you. So that's what we did. And we identified that array SNPs are mostly enriched in enhancers that are active in CD4 positive T cells. And that's why we have been looking at CD4 positive T cells in array. But it will depend on what's the cell type in which your microRNAs are expressed and are active.
Excellent insight. Maybe I'll jump down here a little bit. So another question for you, Gisela. Just read it off for the group here. So you were working with sorted CD4 cells and monocytes, which helped you figure out the different TADs between the two cell types. When analyzing FFP, you have bulk tissue analyzed. What do you think are the main challenges of interpreting bulk tissue Hi-C?
Yeah, so the more we look into cell subpopulations, the more we realize that you have to look at the right cell type to understand how genes are regulated. So even though we have looked at CD4 positive T cells, we now think that that's a very heterogeneous population still. So we are trying to look at more specific T subsets like T regulatory T cells or T follicular helper cells. So it's really important to look at the right cell type, because the regulation of gene expression is highly cell type specific. Obviously, it depends on your application, because if what you want to look at is structural variants, like Kerstin was showing earlier in cancer, then I guess that matters less. I don't know what Christine thinks, but because what I look at is the regulation of gene expression, the cell type matters, but if you are looking at structural variants, it might not be as important. Do you have any comments on that, Christine?
Well, I think for me, we are mostly wanting to look at tumor tissue, and the more tumor, like the higher tumor percentage, the better off we're going to be for identifying some of those things. But yeah, maybe slightly less important, but
yeah.
Okay, great. Another question for you, Kristen, actually, since you're on here, unmuted. Let me scroll back up to it. There's a question about public databases where you can find information of 3D genome annotations to help interpret these variants found in patients with rare diseases. Any help out there in the world?
Well, I mean, there's like 3D genome browser and some tools like that. I don't know if that's quite what you'd be looking for. I know when I'm looking at a lot of like the cancer data sets, we find genes that look like they're of interest, and we're mostly looking at just broad gene databases and disease, like cancer gene functional databases, those kinds of things. So as far as like 3D genome specific annotations, I don't depend on that very much for what I do. But I don't know if Gisela has any other tools or things like this.
There's lots of publicly available data. You know, nowadays we can't publish our findings without making the data accessible to people. So there are lots of data sets that you can access. It might not be exactly the cancer type you're looking into, but there will be lots that you can find. Yeah.
Excellent. The next question actually was a question I was going to put out there to the team. So great to see it. And one of my favorite slides of your talk, Gisela. They first said, thank you for the great talk. And they were interested in the key information to have before deciding to apply Hi-C for disease causal SB analysis. So example, you showed GWAS, and there's other bits of information you can bundle together: GWAS, ATAC-seq, ChIP-seq, RNA-seq, etc. How do you decide, or how did you decide to layer all those bits of information together, and in what order?
Yeah, that's a very interesting question, and it's actually an area of research that people are putting a lot of effort into, to annotate GWAS variants using this wealth of functional genomics data set that we can access nowadays. And I don't know, so what we tend to use is as much data as we possibly can integrate.
Everything.
Yeah, so there's so much data out there. And if you have a very specific research question, if you have a very specific region you want to look at, I would advise that you look at everything. But there are pipelines out there that you can use to annotate your GWAS variants, like FUMA, and there are loads, the priority index, Garfield. There are lots of packages, mainly in R, that you can use. And these packages can pull together all the information that it's publicly available to give you an answer.
Okay, great. Actually, the last part of that question puts you on the spot a little bit. If you couldn't choose all genomic technologies to analyze, maybe you have a little bit of a constrained budget. Could you pick kind of 1, 2, or 3 to use before you would jump into Hi-C?
Before you jump into the Hi-C. So in what context? In the context of interpreting GWAS?
Yes.
If you don't have any money, you're fine, because there's so much data out there that you can use. So use all the data that you can. And then if you have to do one experiment, if you don't have any money, use CRISPR. So use publicly available data sets to generate hypotheses as to what might be the mechanism by which your SNP affect the causal gene. Then use CRISPR to validate that and to see in which direction this effect is going. Is it increasing the expression of the gene? Is it decreasing it? And CRISPR is cheap.
Perfect. Let's see, we are pushing right up to the top of the hour. So let's just have maybe a couple more questions, which there are just a couple more, so that's perfect. Let's see, another one for Gisela, keep you on the hot seat here. So it looks like some questions about some follow-up studies. So they would like to know, are you planning to characterize any SNPs individually? Or the idea of your team, or the idea of your team is to determine the effect of the whole region. In case you plan to characterize an individual SNP, what experiments would you use, and how would you choose that candidate SNP?
Yeah, so I think I just answered the question with the previous one. So yes, obviously you need to validate these enhancer promoter interactions. And what we are using currently is CRISPR. So you can use CRISPR to do a more targeted approach, just interrogating a SNP that you can prioritize putting all the evidence together. Does it have a high posterior probability from GWAS studies? Does it overlap an enhancer in a cell type that is relevant for your disease? Is there an EQTL with that SNP and a specific gene? And so you build up the evidence for a particular SNP, and then you can perform CRISPR to change the SNP, to delete the enhancer, to activate the enhancer, lots of different things you can do with CRISPR. And in terms of interrogating the whole region, yes, you can do a CRISPR screen where you can target all the SNPs that are associated, because it's really difficult to prioritize SNPs sometimes. So you can do a CRISPR screen to target all of them to see what the expression, what's the effect on gene expression afterwards. And that's how we are following up. We are also doing mouse models. So we are deleting these enhancers that we think are important in disease in mice, so we can characterize what happens with gene expression at the whole organism level, but also at the immune system level. We can induce arthritis and see what's the effect of the enhancer deletion in arthritis and so on.
Perfect. You mentioned EQTLs, and there's a question here actually about that. Sounds like it's part of your follow-up studies.
Yes.
Yep. So maybe we don't need to go into too much more detail there. Maybe some future webinars to come. Let's see if there's anything else to go through. Any comments about overlapping GWAS variants? I think that's the last part of this question here. Using EQTL data, helping your SNP to gene targeting.
Yeah, absolutely.
Yeah. So we can maybe
definitely.
skip that one.
Yeah. Tushave, drop me an email if you want to, for Tushave Vianney.
Yes. Yeah, a little shout out. I'm sure he or she can find you on the web, and definitely send you a note or send you a note here and connect about working together in the future. Well, just wanted to say thank you again. I think we'll wrap it up here. We're just right at the top of the hour. Just want to say thank you again. Hopefully everyone's a little bit more excited about the novel insights they can gain from 3D Genomics from today's webinar. Just want to thank Kristen and Gisela for your time. Really appreciate you guys making the trip to Vienna for ESHG, and as well presenting some of the same information here for the rest of the world to see. Thank you all for your time and attendance. We appreciate it, and stay tuned for the next webinar upcoming. Cheers all.